My research started from the area that I spotted there was a huge gap when I was working at industry. And that gap still exists and still is leading me to new areas and opportunities within the area of my research. So if you bear with me, I would like you to join me in this journey. But whilst I'm talking about my journey, I want you to try and see it from my point of view as a PhD student or as somebody who was exploring to join academia again to do her research from years, many years of industry, working in various parts of the industry, from catering in airline industry to new product development and regulatory issues around technicality, labeling in the whole industry of food. So when I started my research, of course, I had a 1-year-old son and I was working in food industry. Jamie Oliver was just coming out. Many of you may be too young to remember any of this, about the quality of the school meals in the schools in the whole UK and how it could be improved. And of course, because I was in an industry that was serving the schools in the main catering, I was tasked to develop new food products. And at that point, although I was successful, I realized that the sales of the company went massively, massively down. And that didn't take me long to work out, yes, the kids don't like the healthier options and versions that I developed because they didn't taste as well. And at the same time, my at the time, actually 6-month-old baby was, I was trying to introduce solid food to him. And he was slightly anemic. And it didn't take me long to realize what was in the market of UK wasn't fit for purpose, because as a mom, I wanted to give the best to my child. I was developing nutritionally optimized or better or healthier quality type food for schools. And how could I then struggle to find a food or any way how to prepare a food which would be suitable for a 6-month-old baby? And Adrian, if you don't mind, we go to the next slide. So, and at that point, obviously, I realized that I need to go back to research. I decided that I gave up my job. I was not fortunate enough to obviously find funding for my research. Many of you might be in the same position as I was. So I was under pressure of bringing up a family, giving up a job and starting funding a PhD into the area of infant nutrition and how we could develop better diet for them or in fact food products. So throughout this session, I'm going to talk a little bit, not too much, I don't want to bore you, around the scientific background, a statement of the problems that I was facing and what is happening currently and the approach I took and in the intervention and my future plans in this area. So next slide, please. So as I said, when I left industry, I was coming into an area where biology, I did almost like an A-level in biology. I didn't do degree in biology. So I had to come back to academia and teach or relearn everything again. By the way, I'm originally Persian, so I'm from Iran, and coming to the UK and starting the study in an English language for me was learning a different language. Remember, talking a different language is different to be able to read and write scientifically in a different language. So I had that to face as well. So I started around biology. And of course, nutrition is a science around biology. And I had to look at it from a life course perspective, because in everything, research is now showing and has been proven that everything starts within the first 1,000 days of life. And infant nutrition, therefore, is the most important. Why? Because if a child from early stage is being either deprived from optimal nutrition, or in fact is exposed to overnutrition in relation to high intake of fat and sugar, they often tend to have a lot of non-communicable disease, such as, for example, diabetes, cardiovascular disease later in life. So that's where I had to go back, learn the life courses and understand the biology of the infant in order to understand and appreciate the nutritional need of an infant. And then, of course, I had to then find the interface between that nutrition and the food that I was used to producing in a manufacturing environment. Next slide, please. So where was the gap? Where was this gap that I was trying to address? I experienced it firsthand as a mom, as someone in industry who was producing better food and healthier food, which was not selling. I was being told off daily by the board. But so where was this gap? It didn't take me long to understand that the gap was in weaning. Why? Because a lot of concentration, even now, was focused on breastfeeding, the importance of breastfeeding, which is quite rightly so. Some of you may be coming from a biology background and an immunology background, so you appreciate that. And of course, a lot of attention because of Jamie Oliver and the media and all around those, marketing was on the school meal. But what we are still, even I believe to this day, we are missing out is the period of weaning, where the child is first being introduced to solid food, where the taste bud is being developed, yes? And the whole food culture and dietary habit is being developed at this important stage of life, when the child has got immunology, which is complete but naive, the immunological system. Kidneys are not as such developed yet, so you can't just go ahead and start giving a type of food that the child at age of 1 or 2 start consuming. And also at the same time, their physiology and their biology is rapidly growing. So speed or velocity of the growth is at the highest rate during this period. Next slide, please. So this is all still my biology. But in order to appreciate this weaning, the gap in the weaning, I had to go down the route of chemistry because I wanted to prove it and I wanted to have data to prove it. Please, next slide. So what did I do? I embarked into the industry, looked at the UK food market at the time, and started trying to come up with a traffic light system. As you can see now in the supermarket is pretty much implemented. I tried to come up with a traffic light system, which is equivalent, but for children under the age of 2, or in fact 1. So I had to come up with development of recommendation, look at the current recommendation by, for example, Food Standard Agency, whether it's by Department of Health, and try to translate that to labeling of food product. And then, of course, do quantitative analysis or chemical, using various methods from or techniques such as HPLC, high performance liquid chromatography, or ICP for analysis of minerals, and some other methods. For example, for protein, I use Caldol. For fat, I use organic solvent method. So by weight difference. So all of these I had to learn and introduce myself to, get the skills in the lab in order to prove scientifically that the current market of the UK was not fit for purpose, both in terms of fat, in terms of sugar, and in terms of salt in many cases. And how could we be designing food for schools where we are missing out on babies? And something I want you to appreciate is when we talk about baby food, because usually infant formula is made by pharmaceutical industries, produced, manufactured in pharmaceutical industry, they come under a different regulation and are very robust. But when we talk about baby food, interestingly enough, they are covered by the same regulations that covers our ready meal in the market. So just think about it. How could this very sensitive period of life not to be in the same category as a pharmaceutical type product, the same way the infant formula milk is? Next slide, please. And of course, then I had to develop index of nutritional quality for these foods. And I wanted to take it one step further, as I said earlier, use HPLC and ICP to look into micronutrients, because I also realized into regulation of the UK, same regulation that looks after our ready meals and fast food, there is no mention of micro and macronutrients such as vitamin A, B, C, and also minerals, as you can see on the screen, magnesium, copper, potassium, zinc. And these are quite important. Why? Because as I said, this is a velocity of the growth in this period, and it's an important period in relation to mental development, as well as other biological and physiological growth. Next slide, please. So I was successful. I proved there was a gap. I proved it needed attention. And I proved this by various scientific publications, being invited in different conferences. And one word of advice, if it is worth anything I can give you all, is usually the research degree committee in your respective universities doesn't require you to publish your work in order to graduate and get your PhD. However, this publication has one benefit. One, gives you the exposure. Secondly, because it's peer-reviewed, it gives you that credibility that you need. And when you go to your final viva, you often tend to find this a much smoother experience, a much easier. And of course, that exposure, I believe that exposure at the time has made my career where I'm sitting here and I'm talking to you. Next slide, please. Thank you. Because of that exposure, then I received international and both nationally and international media attention. Adrian, if you don't mind to press 3 times, these would fly in. Various, newspapers and various organizations, they picked on the data that I reported. And of course, then as a result, I ended up in the media. And even currently now, I am being approached by various TV channels. Yesterday was on the BBC for Inside Factory, for me to go and comment on various food products. Next slide, please. And of course, that led me then to actually write the first UK guideline for infant feeding. And that was published and acknowledged by Caroline Walker Trust, which for me, what I'm trying to present to you here is when you put yourself outside that comfort zone, the opportunities would come. And you need to be able to see the opportunities and spot them. Sometimes it's very easy to be bogged down in a small area and too focused. All the time, try to see the relevance of the research that you're doing within the context of the society and the impact that it might have. I promise you, every single one of you, if you look a bit carefully and try to be creative, you can see a relevance and impact of your research. Next, please, Adrian. And of course, this, all of these guidelines that I wrote only led me to understand that I know very little. And there is a lot more to be learned about feeding, the nutrition of an infant, the portion sizes, frequency, the composition, etc., etc. And you may ask me, no, we've all been fed and we all know what to do. Our parents did it, my sister or my cousin, my family did. Yes, we are all doing it. But one thing I would like to draw your attention is that rise in the childhood obesity that we are witnessing in the UK, and we are still struggling to address it. Despite the sugar tax that we are bringing, despite all the recommendations that we are putting, it seems like we need to go back to product development, and we need to go back to that early stages of life and start rethinking our food culture. Next slide, please. And of course, my focus then from there went to function of lipid, because at that time I realized, okay, I've written the guidelines, we know about vitamins, minerals, but what I wanted to know was, what is the impact of fatty acid? Because you do all appreciate that this issue or this hype we were receiving from fish oils are good for cognitive development, cognitive function, and quite rightly so, visual, and in fact, overall health, maybe even later in adulthood. Next slide, please. And all of this brought me back again to biology. I now had to teach myself biochemistry. So you see, I go back again from something that was supposed to be food. I went to nutrition and biology. Now I'm coming back again to biology to understand the pathways, mechanism of absorption of fatty acid, linoleic acid, as well as alpha-linoleic acid, which is basically your fish oil on the right-hand side. And it didn't take me long to work out that, okay, in infant, fortunately or unfortunately, the pathways are not exactly as adult. And in fact, the conversion from that alpha-linoleic at the top down to DHA is insufficient. And there are 2 times that this delta-6 desaturate enzyme was being used, and that's a regulatory enzyme. And these 2 different pathways are competing. So depending on how much of the linoleic acid, which is your sunflower oil type, you're consuming in your diet, versus how much of fish you are consuming, which is that one at the top, alpha-linoleic, ALA, then it would then be determined how much DHA your body can actually produce. And that's for adult, let alone for infant, that this pathway is going to be insufficient. Next slide, please. But I had to then go away and prove this. Again, I had to read the literature. I had to find out that oily fish was the best source of it. And of course, this conversion rate is very low. And when it comes to DHA, we are looking only at 0.1% respectively. So it's very small amount. Bearing in mind the dietary habit in the UK, remember those foods that I analyzed, remember that we didn't have any guidelines, and most of parents like I didn't know, I couldn't find anything. Next slide, please. So it was very important when I wanted to find evidence to appreciate that, okay, the DHA status actually depends on the diet, how much the child is taking in, and metabolism. And in fact, even up to now, these days, people who are in the field of nutrition, they don't appreciate that you don't breathe in nutrients. You have to intake them and consume them from your food. Therefore, food science and nutrition, in my view, are not separable. And this is something that I think we are not still there. People are working in silo and they're not appreciating this. And of course, then the evidence became more apparent to me that, okay, DHA, the concentration is found higher in the gray matter of the cerebral cortex of the brain. And of course, the visual development of a child also depend on it. And I had to look into see what is the recommendation. Funny enough, no recommendation in the UK. So we didn't have that. So I had to look at EFSA, which is European Food Safety Authority, and they were recommended 100 milligram per day for a child which is between 6 months to 2 years of age. Next slide, please. So I want you to just see it from the journey point of view and how I made decision to push this forward. I digged a bit further, I needed evidence, and I found that, okay, is DHA, that is very important. And of course, we need to have intervention in order to introduce it. Next slide, please, Adrian. And how could I prove this without considering breast milk? So when I looked at breast milk and content of the breast milk, it was quite important because we were really driving and say an infant up to the age of 6 months should be feeding only on breast milk. So when we look at breast milk, we realized actually the quality of the diet is not only important for the infant, the maternal diet is also as important. So what mother was eating as part of their diet had a direct impact on the concentration of the DHA or this fatty acid in the breast milk, as you could see on the screen. Next slide, please. And then a little bit further, we explored and we looked at the mental development index of an infant, and we realized, okay, most of the time we were wrong to think only DHA is important. And in fact, the combination of DHA and arachidonic acid is important. So not only we need sunflower oil, but we also need the fish oil. So if we went only for fish oil, because of those pathways are insufficient and you're competing in those pathways for the same enzyme, you will have a lot of DHA, but not enough AA, which is arachidonic acid. So as you can see on the screen, combined, they had better impact on the mental development of an infant. Next slide, please. Then we looked at the psychomotor development of an infant. The same picture was presenting itself. So we wanted both DHA and arachidonic acid. Next slide, please. And it was important then to look at experts' opinion, and we could see, for example, British Nutrition Foundation, WHO, and American Dietetic Association. They all have different recommendations coming. But this was to do with total daily intake. For me, as a food scientist who wanted to develop a food, I still had a lot of questions to answer. Number one, I need to analyze the food product to see how much is in those food products. Number two, once I prove that, I need to find out how much I need to add. But how do I know how much I need to add if I don't know exactly what should be the intake of an infant on a daily basis? Next slide, please. So again, you can see that I was looking at the same time into regulatory aspect and recommendation aspects between nutrition, interface between nutrition and food. We started the investigation. We went back to chemistry. I used reverse phased high performance liquid chromatography, aerosol detector, and charge aerosol detector to see whether we can detect them. Remember, the limit of detection and limit of quantification in these type of products that you can see on this slide sitting on a supermarket shelf was really, really low. And therefore, it was very difficult. I had to adopt all the quantitative method that was available to me in order to be able to match the matrix of these foods and actually set a limit of quantification good enough for scientific community. One point that is interesting is these food products, they're older than the babies that are going to consume them. Majority of these products are produced more or less about 8 to 12 months before even the baby is born, because they are produced by a process called retort in food manufacturing, and they're sterilized. So they have long shelf life, just like the long shelf life milk we have. Next slide, please. Again, the picture was the same. They were lacking of the fatty acid. At this point, I was obviously an academic. So I finished a PhD, I joined academia, and I was fortunate enough to start with my first PhD student, who is now Dr. Emma Lafril, and followed by Sharon, that is now also Dr. Sharon Thompson. I have to apologize to Sharon. And at this point, I had basically two very bright students who joined me. Again, just something I need to bring to your attention is both Sharon and Emma joined me directly from BSc, and they came through foundation degrees. So what I'm trying to highlight here is that where you come from or what is your academic journey or the background where you started doesn't have any impact on your future as long as you're open to opportunities and you work hard. That's the most important point here again. And they joined me, and I started working on developing systems or methods of fortifying food. But remember, I'm fortifying food for babies who moms wouldn't like to see anything being added to their food. And also, I'm in an area that there is no recommendation. Nobody has done it before. It's a simple concept. They're doing it into the milk. But for me was to introduce it in a way that I can be assured when I'm processing it is not being destroyed. When I'm heating it to sterilize it, is not being destroyed. Next slide, please, Adrian. So yes, daily intake was the hassle for me. As I said, there is no regulation, there was no recommendation, and I had to go back to my standardized menu. I had to come up with some mathematical formula in a way that I could make a parameter setting that would allow me to decide what should be the dosing. I looked at how much a baby intake is from an infant formula, how much recommendation is for amount of milk, because milk is the main diet. Then how much am I identifying in this type of food product, calculate the gastric capacity. These are all taking me back again to biology. And from there, then compare the two, and whatever is left over is going to have to be complemented from the winning type diet. Next slide, please. So as you see, there was a lot of data coming in, and now, in fact, I'm trying to make this almost like a machine learning, trying to turn it into an algorithm that automatically can do this based on personalized nutrition. But imagine at the time when I was coming up with this data, at the time, all that mattered for me was how much do I need to add into the food? So I just did a simple calculation to minus them and come up with the total daily intake based on my standardized menu. The standardized menu, again, was my creativity, and nothing existed out there. All I had to do was to go and look what the manufacturer of the milk is suggesting, what is the recommendation or instruction on those baby food in the market, go and calculate the gastric capacity of a child, and then come back and calculate and just see how much How much do we need to rely on this type of food product? What do these food product has to provide? And as you can see from results that is showing, in terms of arachidonic acid and DHA, they were both below the recommendation. But the top two, yes, the child was getting enough because formula was being fortified. So that again highlighted to me that formula was quite important. And when it comes to breast milk and recommendations are for breastfeeding, then we need to ensure that diet of the mother is just as good, because otherwise the breast milk, as you could see in the earlier slide, was lacking this fatty acid. Next slide, please. So my way of fortification, of course, then was, for some of you who may not be familiar to this area, was encapsulation. And I'm not a formulation scientist. I had to go back and learn about formulation. So now we are leaving nutrition and food, but still keeping them on track, because that's what we need. And that's our target. But I had to learn about encapsulation. I had to learn about spray drying and how we could use polymers. By the way, polymers that are food compatible and biocompatible. They were both as important. Why food compatible? Because if you're in pharmaceutical industry and you're developing a medicine or drugs, they are all dosed and also they are issued by prescription. When you're developing food, it's sitting in the jar, is on the shelf of a supermarket, and there is no way you could control the intake. So I had to make sure I'm not going above the recommended nutrient intake, or in fact above the values that can cause toxicity. Next slide, please, Adrian. So of course, two methods I employed. One was spray drying the fish oil directly. Another one was to form a nano emulsion first and then spray dry it with the same polymer. And to see which one of them can protect it better. That was the whole point, because remember, these foods are sitting in a shelf, on a shelf for a long period, and they have been pasteurized, or in fact they're sterilized. And the matrix of food, you've got pH of various pH, various matrix, and it's important to make sure they're not interacting, or they're actually the integrity of them is actually retained throughout the processing. And then of course take it further, do the analysis, validate that these are working. And this time I was using gas chromatography, and characterize them using various analytical techniques, as you can see on this screen, that would allow me to show the integrity of this compound within the food matrix. Next slide, please. So now we are pretty much in the domain of chemistry. We analyzed them after formulation. We calculated the yield, and we look at the efficiency of the encapsulation. And we could see, for example, a spray drying, as you can see, was giving me better yield and giving me better efficiency. Water activity was okay, the moisture content was all right. And the purpose for me here, as I mentioned earlier, was to also make sure that this is pH dependent, the formula, so it would not release in the stomach, and it will see the compound right into the intestine and only release it in the intestine, because then I could be assured that I can, at least based on theories, be assured that this is giving the maximum level of bioavailability possible from this food. Next slide, please. And of course, then we took it to SEM. SEM allowed me, scanning electron microscopy, allowed me to check whether there is no apparent cracks on the pores, because that would then show whether it has been compromised through the processing and has released the compound. But it was reassuring to see that the compound is intact, and the encapsulated material is basically still existing within the food, and nothing is interacting with each other. It's not being permeable to other compound, and fish oil is well capsulated inside. Next slide, please. And when we looked at it also, we looked at the decreased values within the week, and of course, spray dried decreased less in comparison to the fish oil, as it was added to the food as a control. So that was reassuring for us to see as well. So all the time I was thinking about, how could I prove this? If it is going to the board and it's being examined, how could I prove that this product is doing what it's supposed to be doing? Next slide, please. We took it further. We looked into FTIR. Again, you can see how much chemistry and analytical chemistry came in here, just to check to see the compound, which is the fish oil, there is no interaction between the compound with the polymer, with the directly sprayed fish, because if it did, it would not be any good in terms of the biological value and bioavailability of the compound. And we could see that, okay, there is no interaction. So that was a green light for me to go ahead. Next slide, please. As the pattern you could see is all similar. So we concluded that we were successful to that point. I'm sorry, I'm not seeing the time now because I've lost completely. I hope I'm running a little bit over if I'm correct. Am I correct, Adrian?
Sorry, I was on mute. No, you're fine, Nazi. Yeah, that's all good.
Okay. Sure. So yes, we concluded that it was pH dependent. It was doing what it was supposed to do. So the next step for me was to try emulsion. Why emulsion? Because it was baby food, and I rather use something that I don't have to look into parents' eye and say I've added anything in terms of fortificant. I wanted to be a natural way of delivering a compound into the food system. Next slide, please. So that's where then, of course, I had to learn about physical chemistry. The physical chemistry, developing emulsion, both oil in water and water in oil emulsion, to see which one is better for me. Again, similar method of analysis, ensuring the for example, droplet sizes are okay, they're not aggregating, they're not precipitating, and actually they're not being impacted by gastric lipids. So all of these were basically my mission for the next phase of my study, learning now about physical chemistry. Next slide, please. Of course, we did choose gum arabic, because gum arabic was food compatible and also is more or less in most of the majority of food products, and it is quite considered safe, and regulation would allow me to use it. So we used gum arabic, we used 5% of fish oil, and we stored it at 4 degrees centigrade, but also I had a control in ambient temperature, because some of the food products, as you could see in the pictures earlier, they sit in the ambient room, and parents, they prefer to have the convenience. If they don't have to store a food product, they prefer that. Next slide, please. So we looked into the stability. We looked at the particle sizes using master sizes. We looked at oxidation. This was very important. As you know, fish oil, they have high number of double bond and they're unsaturated. Therefore, oxidation was a huge problem for me, plus shelf life, plus processing temperature, and of course, level of oxygen that was into the processing of this food. Next slide, please. So we looked at both, and as you can see on the right-hand side, when we compared the temperature below 4 degrees with 21, I could see the 4 degrees holding better in relation to the total oxidation value. And 26 is the value that is the maximum level you could allow in food. So I had to keep below. So of course, it was showing me that if I have food that I can store under 4 degrees, which is refrigerated type food, then this emulsion system is much more suitable. Next slide, please. Thank you. We took it to fluorescence imaging. We wanted to see the interaction between the encapsulated material as well as the fish oil. Picture A on the left-hand side of the screen obviously shows the gum arabic, and picture B is showing the fish oil alone, and picture C is combined together. We could see that the integrity of the emulsion system is retained despite the high temperature of processing of the food and sterilization process. Next slide, please. Thank you. And after that, the question was dosing. I have to dose it. And this is not, as I said, a medication that you could easily dose. And because it's based on prescription, it's a food that is sitting in the market and people can have as many as they want. So I had to look into, again, biological values, how much is the nutritional recommendation, and look into how much in a per jar of food or gastric capacity of a child, price it against the value of the food, but the affordability is important here. So when you're doing this, I had to consider the economy. as well. And the home economy and affordability is quite an important, and as well as regulatory issues. So we designed it, and we used it in the food. And next slide, please, Adrian. We concluded that, yes, we could have these emulsion systems. They are improved bioavailability, and they have improved stability if we can keep them in the temperatures that are relatively refrigerated or fresh range of type of food. And for that, of course, then I had to consider, rather than sterilizing the food, using high pressure processing. And that is a relatively new technique where we don't use heat to pasteurize food, and we are using hydrostatic pressure in order to pasteurize the food. Gives you about 31 days shelf life, and you have to keep it in refrigerators, but it is useful because you can be assured that integrity of the nutrients, whether it's vitamins or whether it's compounds such as what we have developed, are retained and are not being compromised, and the baby basically receive what they deserve to receive for that period of life that they're entitled to. Next slide, please. Now, future work, I need to prove that this is safe, because obviously adding something to a food that is not sterilized has got microbiological consideration. And I also have to consider reducing the amount per day, because sometimes it's very difficult to feed a child 7 to 9 months. Those of you who have experienced this, you can't always be sure that they have the whole food or whole portion size. So we need to reduce the portion size. And also we need to consider addition of alternative alpha-linolenic acid, which is coming from algae oil, for this interest in relation to sustainability of the food, and equally for parents who they wish to have alternative vegan range of type diet. Next slide. I believe, yeah, thank you for listening, and I'm sorry for the little bit of disruption we had at the beginning. If you have any question, I'm more than happy to answer them. The quick overview of the journey I've been and how much I have maneuvered between different sciences, teaching myself. You know, I had to teach myself. If I could, you all can, because you're all younger than when I started.
In fact, that was going to be one of my first questions before throwing it open for everyone else, yeah, was to ask you perhaps to just say 1 or 2 words about how you went about getting the training and networking and everything else, because it was clear from what you said, you've got certain expertise, you needed to get certain other things like physical chemistry techniques and so on. And absolutely. So for all the ECRs here, I wonder if you could just say something about, how did you network and how did you make those connections and so on?
Yes, very important question, Adrian, very good. Number one, it's very important you are willing. You have to be willing. I was willing to take the challenge on. Yes, there was no other way. Yes, remember, I left the job, I'm paying for it, and I'm stuck. I had to find a way. So I was willing to learn. 2, I did network. Yes, I didn't reinvent the wheel. And I tell you what, when I started, Adrian would appreciate this. I had 2 supervisors from chemistry, 2 professors from chemistry and analytical chemistry. When I said to everybody, I'm doing a PhD in baby food, for example, it raised eyebrows. I'm telling you, it did raise eyebrows. They were developing drugs, cancer research, you doing baby food. But you see, now it's such a hot topic. It's all over the media and it's all over the research areas, and a lot is being invested in food. So 2, I was willing to network, and I didn't reinvent the wheel. Where I could rely on people to do something for me, I did listen. I didn't leave it on the shoulder. I engaged, but they did it with me. And in return, because I was publishing, I acknowledged their contribution, you see? So I had something to offer in return. That's the best way to go about collaboration. Collaboration is about give and take. Yes, it's an exchange is happening. It's almost like a business. So I worked at that very quickly from the start, but also they taught me how to do it. Remember, I couldn't have not become expert within the short period. You all understand what I mean, because you're doing a PhD, you have limited amount of time and resources, plus money. So how could you do everything? You can't. It's best if you can collaborate and network, and in return, see what could you offer them in return. And if you have got a mindset of, I want to be impactful, whether it's publication, whether it's attending conferences, and you are going to acknowledge them, that's the best return you could give them at that point. So collaborate and be open-minded. Yes, if you are in the area of chemistry, it doesn't mean you can't work with biologists. You're in the area of biology, you can't, there is no point to say or reason you can't work with chemists or formulation scientists, or in fact pharmacists. Now I'm looking into personalized nutrition. I need pharmacists. I'm looking at, and in fact, I'm talking now to mathematicians because I need machine learning and algorithm. I can't learn it all. I'm not much older as well. My brain is not going to support me, but I will do that through collaboration. And that's the honest, true opinion of mine. The best advice I think I could give anybody is that.
Brilliant, thank you. There's a question in the chat from Cristiano. Children obesity is obviously still rising, and that the issue isn't solved completely yet, of course. So I think perhaps he's thinking about his future career as well. Are you closer to solving the problem when you were working in a company and the opportunities offered by industry, or do you think you can make better progress inside academia?
Okay, very good, Cristiano. As a result of me being in academia, correct? And I'll tell you why I stayed in academia. I am now parliamentarian, so I'm influencing policy. Only last July, I gave a talk to Parliament, yes? And I demonstrated to them that there is no point you are trying to tackle obesity if you're not considering babies and you're not coming back and tightening the regulation of baby food in this market in the UK. But you're absolutely right. I think both similar. My reason to stay in academia was I always wanted to have I was motivated by money. I'm being honest with you. I wanted to have my own baby food company. Yes. I had very VCs around, venture capitalists. They were around, they wanted to invest. And I stayed in academia to develop that, to take it to market. Correct. So maybe if I had that success, I could have been the pioneer in this area. And I am working with the first fresh baby food in the whole Europe and UK market. Yes. So I have been to a degree successful, but it takes the mindset change. That's why I thought policy is better. We need to go back to government. We need to change from early stage. So my answer to you would be both. Reason I'm saying both, because I haven't disconnected myself from industry. As Adrian said, I'm enterprise lead for a reason. I'm talking to all SMEs in various areas, not only baby food. And I'm constantly trying to look for impact. That's the whole point. There is no point to do research if you're in the lab in a box. Think about the impact and take it out. For me, the impact is food industry. For you, even if you're developing the minute or a small amount of a compound that is going to be used in a drug somewhere, sometime, there is still an industry that you are being impactful. Keep your connection with that industry because that increases your job prospects as well. So keep your contact, learn what are the challenges they are facing and try to address those challenges within your research. And remember, research evolves. I showed you my journey. You know, I jumped from, it's like a monkey. I jumped from one to another, but I was telling this story. They were not disconnected. I was constantly maneuvering and changing, but it was within the same framework. I'm not suggesting to you to change constantly. No, stick to it. But you can maneuver around and try to increase your impact. I hope I'm answering you, Cristiano.
Oh yes, yes. I'm in the same mindset as you, because before joining my PhD, I was 4 years in a food company. And I know how hard it is to talk to CEOs, even though you have the data explaining you are doing this wrong or we can do this better. It is impossible. So it is a long way, a very long way, and knowledge is not enough for sure. We have to do more.
We have to
communicate, to policy making. interdisciplinarity, collaborative projects. Yeah, well.
Absolutely, absolutely. I totally agree with you. Yes, in terms of policies, but equally, in fact, engineering. I need engineering because this hydrostatic pressure processing, it's not something food scientists did. It's not something that chemists did or biologists did. This is food engineers who have come with this idea, and they've developed a way that we could actually process food, pasteurize it without any heat. That's amazing. You will have a strawberry that has been processed and it hasn't changed color at all. You don't need preservative, you don't need additives. It's fresh, the odor, the flavor, everything is there. So to me, that's food engineering. So you can see that how multidisciplinary a topic can be. Yes.
Great, there's lots of other comments in the chat.
Yeah,
Nazi, have a look when we finish in a minute, saying this is excellent, brilliant, very well said, Nazi, and so on. So you're getting a large number of very positive comments.
Yes, thank you very much.
Anyone else got any other questions? If you'd like to put your hand up or type it.
I just have a comment, really. The one sentence that impacted me the most from your presentation is that the baby food was older than the babies, and the shock horror. Oh my God, I hadn't thought about that at all.
No, the baby food in this market is older than babies. And I have another phrase that I love is the quality of our pet food in this country. I'm talking about nutrient density. Quantitative chemistry analysis proves it. Nutrient density, gram per 100 gram or milligram per gram, proves is much more poorer than the pet food. Because pet food, under a different regulation, are allowed to fortify. We are not allowed to fortify. Yet, it's the same regulation as adult meal. So don't fortify it, but equally process it to such a high temperature and leave it for 2 years on the shelf. It's quite sad.
I was going to ask the slightly controversial question, which I guess is the obvious one to follow that up with, is A, why is that in this country? B, is that an EU regulation? And C, what effect is Brexit going to have on the change in EU regulations?
That's right. That's right. In fact, this is interesting because, to be honest with you, the regulations are UK-based and EU adopted from us. So that's it. To what concerns me, if we are leaving EU, I mean, we have left the EU, and now they are looking into revamping the whole regulation. And to me, there could be a danger in order to accommodate import from US and Australia and other parts of the world that they've got much, less robust regulations than EU. What are we being exposed to? You know? And the third one, the problem with baby food is across the world. And in fact, it's worse in places like China. But US is slightly ahead of us because this fresh range of baby food already has been happening in US. But yes, everywhere. Where you see obesity, I think there is problem with feeding infants because that's where taste bud is developing. That's where whole food culture is developing.
Fascinating. We've got about 2 minutes left. So any final comments or questions for Nazi?
I just want to say thank you from the DTA for coming and
joining.
joining. And
thank you for inviting me. Thank you for inviting me and listening.
Let's all give Nazi a huge virtual round of applause, shall we?
Thank you very much.
Thank you.
Appreciate that. Thank you.
Things back to Jenny now, is it?
Yes, that's right. Thank you. Thank you, Adrian. Thank you, Nazanin.